Hello everyone, today we will discuss about cell injury. There are two types of cell injury, that is reversible cell injury and irreversible cell injury. Reversible cell injury occur due to energy dependent ion pumps failure. There is energy dependent ion pumps failure, and then there is inability to maintain ionic and fluid homeostasis in the cell. Therefore, what happens is water goes inside the cell and the cell cytoplasm of the cell as well as organelles become swelling. There are two main morphological features of reversible injury. They are cellular swelling and fatty changes. Cellular swelling occur. Cellular swelling occur in associated with increased permeability of the plasma membrane, while these fatty changes occur due to appearance of vacuoles where the triglyceride was contained initially. There are several microscopic features of cellular swelling, and they are: in the cytoplasm you can see small clear vacuoles, and also there are distended and pinched off segments in the endoplasmic reticulum. Other than these two main morphologies, there are other morphologies as well. You can see, I'll show you in a picture. The other changes you can see from this first picture, there is bleb formation in the membrane, blebbing, blunting, and distortion of microvilli, and loosening of intracellular attachments from the plasma membrane. Then the other change is the mitochondrial swelling. Here you can see swelling of mitochondria. Here these are the mitochondria, these are the endoplasmic reticulum. And then there is a detachment of ribosomes from the endoplasmic reticulum. Then the other one is clumping of chromatin in the nucleus. In this nucleus there is clumping of chromatins. Then what are the two main features of irreversible cell injury? They are cellular swelling and fatty changes. Then the other changes are membrane blebs formation, mitochondrial swelling, dilation of endoplasmic reticulum, and detachment of ribosomes, and clumping of chromatins. Then we will have a look on irreversible cell injury. There are two types of irreversible cell injury: necrosis and apoptosis. In the necrosis, there is it is a form of cell death in which cellular membranes fall apart and cellular enzymes will leak out of the cell, ultimately causing digestion of the cell. In apoptosis means it is a pathway of cell death in which activated enzymes degrades the cell's own nuclear DNA, and there is nuclear cytoplasmic proteins. What happens in apoptosis? There is activated enzymes, and those enzymes degrades cell's own nuclear DNA and nuclear cytoplasmic proteins. In necrosis, there are three main morphological changes in cytoplasm, nuclear changes, and fate of necrotic cells. There are three cytoplasmic changes, and they are: first one, increase eosinophil count. Then the second one is glassy homogeneous appearance. This glassy homogeneous appearance is due to loss of glycogen protein particles. Then the third one is there is enzymatic digestion of cytoplasmic organelles. In nuclear changes, there are four types of changes. They are pycnosis, which is characterized by nuclear shrinkage. Second one is karyorrhexis, which is fragmentation of the nucleus. Third one is karyolysis, in which basophilia fades because of digestion of DNA by deoxyribonuclease activity. Karyolysis. Finally, there is nucleus death and disappearance of the nucleus. When we talk about the fate of necrotic cells, there are two pathways. Dead cells can be replaced by formation of myelin figures, or else sometimes can be become calcification. Then we will discuss about types of necrosis. There are 6 types of necrosis. They are coagulative necrosis, liquefactive necrosis, gangrenous necrosis, caseous necrosis, fat necrosis, and finally fibrinoid necrosis. Here in this picture you can see coagulative necrosis of kidney. Here is a kidney and here you can see coagulative type necrosis. In coagulative type necrosis, during first few days, although the tissue is dead, underlying tissue architecture is preserved for few days. Then finally denature of proteins and enzymes and form eosinophilic anucleate cells. This coagulative type of necrosis you can see in almost all solid organs except in brain. Then here is liquefactive necrosis in brain. I told you earlier, you cannot see coagulative necrosis in brain. Therefore, you can only see liquefactive necrosis in brain. In brain, liquefactive necrosis occur. Here you can see liquefactive necrosis. It is occur due to accumulation of inflammatory cells. Finally, those dead cells are digested by phagocytosis and this tissue become viscous liquid. In a bacterial infection, you can see creamy yellow pus formation in the brain. In the first picture, you can see caseous necrosis of lung. Usually caseous necrosis can be seen in tuberculosis. You can see this appearance is like cottage cheese like appearance. Microscopic appearance of this area is it is an amorphous eosinophilic granular appearance. Amorphous means there is no morphology. Eosinophilia, and due to that it is represented by pinkish color appearance. Here in the second picture you can see fibrinoid necrosis in an artery. Usually fibrinoid necrosis occur due to immune reactions, and those immune complexes and plasma proteins get deposited in the walls of the vessels, producing bright pink amorphous appearance due to fibrin deposition. Here in this picture you can see fat necrosis of pancreas. In fat necrosis, there is focal areas of fat destruction. It is typically resulting from release of activated pancreatic lipase into pancreas and peritoneal cavity. You know there are fat in pancreas as well as in peritoneal cavity. After releasing this activated pancreatic lipase enzyme to pancreas and peritoneal cavity, fatty acids are released. Then those fatty acids are combined with calcium and produce grossly visible chalky white areas. You can see grossly visible chalky white areas here. Finally, the other necrosis type is gangrenous necrosis. Actually, this gangrenous necrosis is not a distinctive pattern of cell death, but, but the term is still commonly used in clinical practice. This may be due to ischemia with superadded infection, usually anaerobic infections, and there is foul smelling and blackish appearance. From this diagram, we will compare the differences between necrosis and apoptosis. When we take the cell size, in necrosis, you can see there is enlarged due to cellular swelling. In apoptosis, you can see there is a shrinkage or reduce the cell size. In necrosis, the nucleus is pycnosis, karyorrhexis, and karyolysis. But in apoptosis, there is a fragmentation into nucleosome size fragments. You cannot see that appearance here. In necrosis, as you can see here, the plasma membrane is disrupted, but in apoptosis, usually plasma membrane is intact. But there is altered structure, especially orientation of lipids. Cellular contents in necrosis may leak out of the cell due to enzymatic digestion, but usually in apoptosis, cellular contents are intact. In necrosis, there is frequently inflammation can occur in necrosis, but we cannot see inflammatory reaction in apoptosis. Necrosis is always pathological, but apoptosis can be physiological as well as pathological. Hope you guys gain some idea about cell injury. Thank you for watching.